Hello, grade 12 students. In this video, we're going to talk about the semester 1 exam review worksheet. The topics that are covered in this exam, they are chapter 1, document 3, 5, and 6, pages 18 to 31. Chapter 15 about the sexual cycle, documents 1, 2, 3, and 4, pages 194 to 202. Chapter 2, documents 1 and 2, pages 42 to 45, and also chapter 3, documents 2, 4, and 5, pages 60 to 69. Also, you have to review the bi-weekly review worksheets plus the notes on your topic. Please keep your worksheet in front of you in order to start to solve. Question number 1, talking about the sickle cell anemia. Sickle cell anemia, Mr. and Mrs. X have a daughter suffering from sickle cell anemia. Document 1. This hereditary sickness, whose mode of transmission is autosomal recessive, is characterized by an abnormality in the beta globin molecule, which leads to the deformation of the red blood cells. Mrs. X is pregnant, and the couple demand prenatal diagnosis, so they have to know if their second child will be affected by sickle cell anemia. By looking into the pedigree, Mr. and Mrs. X, they are normal. So they are normal, but they have affected child. So the parents, they carry the allele of the disease, but it was masked. So they are heterozygous and they have the normal phenotype. So let N capital be the symbol of the normal allele, which is dominant of the allele of the disease, which is recessive. And let take S small, since it's sickle cell anemia, with the symbol of the allele coding for sickle cell anemia, which is recessive with respect to the normal allele. And here you have to find out the genotype of the daughter. The daughter have sickle cell anemia. So it should be homozygous since the allele of the disease is recessive, having the genotype as small. While the parents, they should be heterozygous since they are normal and they gave birth to affected daughter. So this daughter will inherit S from each of their parents. So the parents, Mr. X and Mrs. X, there will be an S. Based on the logical reasoning, find the probability to have an affected child. What is the probability to have an affected child? So having an affected child, both parents they are heterozygous. So half of their gametes will be to have the allele of the disease. So the probability for this couple to have six children is 1 over 2 times 1 over 2. So it's 1 over 4. Document 2, it reveals the sequence of the parts of the non-transcribed, pay attention, non-transcribed strand of the beta globin alleles. HbA is the normal and HbS is the abnormal, is the muted one of the beta globin gene responsible for sickle cell anemia. A direct diagnostic method by radioactive probe is done for this family. Many copies of the parts of the beta globin gene can be obtained from the DNA of each person by this technique. These copies, they are separated into two lots, and each lot is placed in a different radioactive probe. Document 3, each probe is capable to bind with either HLA, allele, HBA, or HBS. The results are shown in document 4. So here we have the document 2 showing the position of the nucleotide for HBA and HBS. So what do they want from you? The part C, specify based on document 2 the location and on the mutation and its type. The mutation mainly is located at position number 7, where it's A and the normal and it's substituted by T. So it's adenine in the normal and it's substituted by thymine in the abnormal one. So this is the type of mutation. This mutation is substitution by point mutation by substitution. For D, determine a reference to documents 2 and 3. So you have to look to documents 2 and 3, which allele correspond to each probe used. If you want to look to the documents, these documents 2 and 3, we have to know that the radioactive probe, it binds to part of the allele by complementing with the nitrogenous bases. So probe number 1, what is the complementary of the probe number 1? So probe number 1, G, the complementary is C, A is T. GHC, and AHC, and so on. This complementary DNA is that of the HBS, is the same as that of the HBS. Thus, probe 1, it permits to find out the mutant, while probe 2 is going to be for the normal. Part E. Do the results of document 4 confirm the hypothesis or to confirm the genotype you have indicated in question A? Justify the answer and draw out the genotype and the phenotype of the fetus. Here, if you like document 4, all of you pay attention to document 4. Mr. X and Mrs. X, both of them, they have probe 1 and probe 2. We said that probe 1 is for the abnormal. So they carry. So the two parents will have the two probes, they are visualized, which confirm that the parents are homo or hetero, having two probes. So they have HbA and HbS, so they are heterozygous. While the daughter, it has only probe 1, so it has a thick band for the probe 1. So the daughter is only corresponding for the mutant, which confirmed that is homozygous and having the genotype SS. While the DNA of the fetus, it does not permit to visualize except for the probe 2. So this corresponds to the allele N, but the fetus will be normal, having genotype NN. Justify why prenatal diagnosis is more accurate than a pedigree in detecting a genetic disease. It's more accurate because it depends on the gene itself and also depends on the real genotype. The pedigree permits to detect the phenotype and the possible genotype. Maybe it's going to be effective or not. So prenatal diagnosis, it's more accurate and it's more precise. Question number 2, determination of the ovulation. First phase of the menstrual cycle is marked by important development of the follicles. Out of these follicles, only one becomes mature and ready for the ovulation. In order to better understand the factor and the mechanism that cause ovulation to occur, the following studies are performed. We have study number 1, the variation of the level of estradiol, ovarian hormone, is monitored during a sexual cycle. The results are shown in document 1. We said, draw the curve, which represent the variation of the estradiol and the function of time. So the time is going to be down and the level of estradiol will be up. You have to draw the graph using a pencil. You have to eliminate the values that you didn't use them, so you have to use a specific scale and you have to give it a title. Draw the curve. We draw the curve. Document 2 shows the variation of the diameter of the cavitary follicle during maturation until ovulation. You have to know that the diameter of the follicle is proportional to the number of follicular cells. First, they said define ovulation. This ovulation is the release of oocyte 2 from the mature ruptured Graafian follicle into the oviduct, into the fallopian tube. 3. Explain how the transformation of the follicle in document 2 leads to the variation of the level of estradiol during the follicular phase. You have to know that explanation, you have to use your knowledge also. For the increase in diameter from 5 to 0.5 centimeter at day 2 to follicular phase for sure to 20 millimeter at the same phase is followed by increase in the number of follicular cells. The number of follicular cells is increasing. Okay, so here you can know that as the number of cells increases, the estradiol level increases from 60 picogram till about 240 at day 12, as shown in document 1. Okay, document 3 shows the follicle at two different stages and the development in the ovary during a sexual cycle. Number 4, we want you to know that name the follicle represented in each of the photos A and B. A represent which one? It represents the tertiary or the cavitary follicle. Since it's 10 millimeter, so this diameter it corresponds to the day at which the follicle is 10 millimeter at day 6 of the cycle during its development, while the document B, it's at diameter 20. So diameter 20, it's just before the ovulation. So it prepares itself at day 12. So it's the Graafian follicle. So you have to use the documents and you have to be careful for the documents. Study 2, two lots A and B of female monkeys. They are subjected to ablation of their ovaries. They receive continuous injection of moderate levels of estradiol which keeps LH at a low level. The monkeys, they are, uh, the monkeys here are receiving a unique injection of high dose estradiol on a specific day. The variation is monitored in these monkeys and the results are shown in document 4. These two monkeys, they are overoptimized. They have ablation of the ovaries. So in the both overoptimized female monkeys of blood air interpret is analyzed plus indication. So we have peak of LH of 16 nanogram per milliliter at day 12. And we have the day 17 for the females are subjected respectively at day 9 of lot A and day 14 in lot B to unique injection of high dose of estradiol. However, this level of LH is constantly maintained about 3 nanogram per milliliter following the injection of continuous moderate level of estradiol. This means that high quantity of estradiol favors the peak of the LH. Already we know this before. 6. Explain that the stimulus leading to the ovulation is given by the ovarian. So the ovaries secrete high concentration of estradiol at the level of the threshold. This will stimulate by a positive feedback the pituitary gland, and hence the peak of LH is shown for ovulation. Moreover, the follicle undergoing maturation emits a stimulus high dose of estradiol that favors its rupturing corresponding to the ovulation. Moving to question number 3, talking about the cancer and genetics. Billions of cells of the organism, they have a limited lifespan, are continuously renewed due to cellular divisions controlled by a system of regulation. The dysfunction of this system of regulation can produce a clone of cells, thus forming a tumor. This later is benign as long as it's controlled, but can evolve into a malignant tumor, which is called the cancer. The cancerous cells lose their contact with the neighboring cells. They tend to migrate and colonize in other tissue. This is what's known by metastasis. Pick out from the text the cause of the appearance of the tumor. So the cause is dysfunction of the system of regulation can produce clone of cells, thus forming a tumor. Definition of the metastasis: whenever the cancer cells they lose their contact with the neighboring cells, they tend to migrate and colonize in other tissues. This is what's known by metastasis. In order to better understand the origin of this type of cancer, several studies have been carried out on the gene p53 coding for the protein p53. This protein intervenes in the regulation of the cell divisions. Study 1. The development of this type of cancer is studied in 3 lots of mice as a function of their genotype concerning the gene p53. 2 alleles, p53 plus, which is the normal, and p53 minus, which is the mutated, are only considered. These studies are shown on document 1, the result of this study. So interpret. So you have to know in lot A have a genotype p53 plus, p53 plus. The percentage of the mice that do not develop cancer is 100%, and remains constant during the 500 days. However, the percentage decreases from 100 to about 70%, where the mice of lot B having genotype p53 plus and p53 minus, while between the days short, between the days 280 or 300 and the day 500. This indicates what? This indicates that the allele p53 minus, it favors the development of the cancer when it's present in one copy. Whereas, let's say having genotype p53 minus, the percentage of mice not developing cancer begins to diminish from 100% on day 50 to about 0 or null on day 250 days, which is less than that day 280 correspond to the appearance of the cancer and to lot B. So therefore, allele 53 accelerates the appearance of cancer and its action is amplified whenever it's present in the homozygous in 2 copies. Document 2 show the nucleotide sequence of the non-transcribed. So specify the type of mutation. If you look at these, the original is substitution. So all of them, they are identical. GGC, 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 ATG, ATC, ATG, sorry, AAC, AAC, CGG, CGG, AGG, AGT. So we have one of the nucleotide is substituted at the position 249, where it's G. In the normal, it's substituted by T, and the abnormal, which is allele 253 minus. So explain how the modification in the nucleotide sequence can cause the appearance of this type of cancer. So this will lead to a specific another amino acid. So this will amino acid different than that was for the normal. This will lead to the abnormal functional protein, and these cells will produce cells that are going to be tumor. Moving now to part 5. Part 5, study 2. Researchers have studied the mutation detected in three groups of individuals. Individuals of group 1, they are non-smokers and non-alcohol consumers. Those of group 2, they are smokers but not alcohol consumers. And group 3, they are smokers and they are alcohol consumers. The results are shown in document 3. They want us to show that consumption of tobacco is the risk factor for cancer. So document 3 that show that in the smoker, the number of mutation by substitution is 5, while the number of individual affected by cancer is going to be moderate. The non-smoker present 3, so 3 less than 5 mutation by substitution that will limit the development of cancer. So this means that between the smokers and the non-smokers, smokers they increase the chance of the mutation and thus increase the chance of having cancer. Part 6 justify that the high number of individuals affected by cancer in the case of the simultaneous consumption of alcohol and tobacco. In both tobacco and alcohol consumers, the number of mutation is 7. The value greater than 5 and greater than 3. So this means that alcohol consumption and the tobacco consumption will increase both of them and will lead to mutations as substitution, deletion, and insertion. So will lead to higher risk for cancer. Question number 4, functional relationship between the ovaries and the uterus. So in the framework of studying the functional relationship between the ovaries and the uterus, many experiments are performed on female mammals. Experiment 1, we inject increasing amount of estradiol. So the results are shown in document 1 at puberty. And experiment 2, we inject only physiological doses of progesterone to an optimized female mass. No significant changes were observed at the level of the uterus. In other optimized females, 0.0 microgram of estradiol are going to be injected, followed by injection of the same previous doses of progesterone. So we observe more amplified results than those presented in document 1. So let's move to document 1. For document 1, what did they say? The average mass of the uterus, and we have the amount of injected. So the average mass of the uterus without injection of estradiol was 12 milligram. After the injection of estradiol, as we increase the injection, the mass of the uterus is going to increase. So the estradiol will stimulate the development of the uterus. While in experiment 2, what did they do? No significant changes were observed at the level of the uterus after the injection of progesterone only. To have only injection of progesterone. So the injection of progesterone has no effect. So it also enhances the development of the uterus and the presence of estradiol in the overoptimized female. What can we draw from this experiment? Justify your answer. You can draw that the estradiol has target cell as the nuclei of the uterine cells. So that can choose all the grains of radioactive estradiol in the nuclei of the uterine cells after the injection of the tritium labeled estradiol. Moving now to part 3. Part 3, experiment 4, what did they do? They inject estradiol to optimize drag at time 0. Then we measure the rate of certain constituents of the uterine mucosa, and the results are presented in document 3 and 4. Knowing that development of the uterine mucosa is related to the mitosis, determined using document 3 and the acquired knowledge, the mode of action of estradiol. So you know that after the injection of estradiol to optimize drag, the increase of mRNA per uterine cell elevates from 0 to 100. So this is the phase of DNA transcription into the mRNA. And the increase of proteins per uterine cell will elevate from 0 to about 60% after the delay of 12 hours. This could correspond to the translation from the mRNA that follows the transcription. So estradiol stimulates the expression of certain genes for the synthesis of protein. So injection elevates the DNA per uterine cell from 0 to 60 after a more delay of 24 hours, indicating the replication of DNA, which precedes the cell division corresponding to the cellular proliferation. Explain based on document 4 the results of experiment 2. Document 4 shows us that increase in the progesterone receptor in thousands of sites per uterine cell from 6 to 5 to 6 to 50 between days 0 to 1 day after the injection of estradiol. They show that estradiol favors the production of the progesterone receptor. Therefore, when injecting progesterone alone in experiment 2, there was few progesterone receptor in uterine cell, and therefore this hormone has no effect on these cells. But after injection of estradiol, the number of the receptor increases for progesterone, and the fixation of progesterone on the receptor increases, thus modifying the cellular function and favoring the production of proteins, which explains the increase in the mass of the uterus. Thank you.